Hi guys, we are back with our today's questions. We have molecular basis of inheritance, some DNA, some RNA and some proteins. So the first one, it says how far in the each base pair from the next one in a double helical DNA model of Watson and Crick, right? So when you're talking about a typical DNA double helix, they have asked that how far is the two nucleotides, the adjacent nucleotides are from each other. Now we know that they are 3.4 angstrom away from each other, and therefore the 10 nucleotides, when they take a turn, we say that is 34 angstrom. So here it is in nanometer, so it is 0.34 nanometer. So remember, 2 nanometer, it is the what you call as the diameter, we say of that particular DNA, which is 20 angstrom. So don't get confused with these numbers. Between the adjacent nucleotides, the distance for sure is 0.34 nanometers. Okay. So the next one, it says the gene expression, it may be regulated at the level of. We know that depending on the situations, the expression of the gene is regulated. So whether that DNA will be converted to mRNA, whether that mRNA will be what you call as translated to a protein. Once that protein is found, whether that will function or not. So at any of these levels, that is at the level of transcription, how it is being spliced, it is being cut and it is being joined, removal of introns and joining of the exons, that is done. So there also you can control it. You can also control at the level of translation. So you can control at all the three levels. So the gene expression, it can be controlled at all the levels of the expression. The third one, it says which of the following amino acids are coded only by a single codon, right? We know that amino acids, they are coded by more than one codons, right? And therefore probably we say the genetic code is degenerate, right? But there are certain amino acids which are coded only by one codon. So two of the amino acids which are coded only by single codons are your tryptophan and your methionine. So the two amino acids coded by single codon, they are tryptophan and methionine, single codon each, I mean, right? Tryptophan coded by only one codon, methionine coded by only one codon. The last one, it says in a typical operon model, the RNA polymerase binds to. Easier one, we know that RNA polymerase, when it binds the DNA, it binds at the promoter and then it starts what you call as its transcription. So in a typical operon model, the RNA polymerase, it binds at the promoter, right? We know operator is the site where your repressor and all, they go and bind. Repressors and inducers, these are molecules which come from outside and they influence the expression. So a typical RNA polymerase in a typical operon model, it binds at the promoter. Stay tuned with us for more such questions with me, Girish Kukreja, on this channel. Thank you.